Alright everyone, now let's move on to systems microbiology. This is a nice section in first aid because it accounts for all the bugs, viruses, fungi, and parasites and organizes them by disease. During our entire review this far, we have learned about each individual bacteria or virus, the characteristics of them, and really what disease they cause. Now we're going to flip that on its head and take a look at each organ system individually and understand how the bad guys affect them. I hope that this will be a nice review and help you understand microbiology from a different lens and give you a big picture of how it all fits together. We'll start by looking at the normal human flora. But first, what is normal flora? Well, it is important to understand that our body has a very interesting relationship with bacteria. I know we have spent a lot of time talking so far about how dangerous bacteria are for us and the serious diseases they can cause. But a cool thing about our body's physiology is that we actually rely heavily on bacteria. We have what is called a symbiotic relationship with them. This basically just means that we both help each other out in different ways. They need us as much as we need them. This is a good thing because our bodies are full of bacteria. Our skin, mouth, nose, and gut are absolutely filled with them. In fact, we have more bacterial cells in our bodies than we have cells of our own. But why are they so important to us? Well, for starters, they protect us from disease. They do this by competing with other bacteria for space and nutrients. And two, they actually produce bactericidal compounds. These compounds can actually kill the other pathogenic or invasive bacteria. These flora are also important in our GI tract because they help us to metabolize compounds. For example, do you remember which vitamin relies on bacteria to aid in its metabolism and proper absorption? That would be vitamin K. Very good. And do you remember what vitamin K is important for? It is important for the gamma carboxylation of coag factors. Do you remember which coagulation factors are gamma carboxylated? 10, 9, 7, 2, protein C, and protein S. Very good. So without bacteria in our gut, we would be poorly absorbing vitamin K and we could suffer from bad bleeding problems. So these are just some examples of why we need bacteria. The sum of all normal bacteria is what we call our microbial flora. So to reiterate, these are bacteria that are found in our bodies. They're found in our bodies on a semi-permanent basis, and they do not cause disease. But why do we care? Well, sometimes these normal flora can get into and contaminate our patients' lab specimens. It is important to recognize that these are just contaminants so that we don't go start treating as if there was a bad infection when in fact it's really just a normal flora that's contaminating a specimen. Also, some bacteria that live in one place can get into another part and cause disease. So for example, can anyone remember what is the most common cause of urinary tract infections in females? The bacteria that most commonly causes UTIs in females is actually E. coli. And as we'll learn in just a moment, E. coli is a part of the normal flora that's found in the colon. And actually it is able to migrate from the colon to the ureter, go up the ureter into the urinary bladder and cause a painful cystitis or infection of the bladder. So this is just an example of how normal flora can actually cause disease in certain situations. So now let's go over some important normal bacterial flora. Okay. Now let's go over some locations and the normal flora that is found in each of these locations. So first, on the skin, Staph epidermidis is normal flora. It is also found in the nose, along with Staphylococcus aureus. Now, thinking back to your basic microbiology, what would you see on gram stain of these two bacteria? You would see something like this, gram-positive cocci in clusters. Now, what lab tests could you do that would differentiate between Staph epidermidis and Staph aureus? Remember, you could perform the coagulase test. Very good. Now, Staph aureus is coagulase positive, while Staph epidermidis is coagulase negative. Very good. So this image is actually of Staph epidermidis. And like I said earlier, these bugs colonize our nose and skin. This is not the same as being infected. In the oropharynx, we can find viridin strep. And in dental plaques, we often can see Streptococcus mutans. Thinking back to strep viridans for a second, what serious disease can be caused by this bacteria? This can cause a subacute endocarditis. Very good. You might see a question on the boards about a patient getting a dental procedure and then having a fever and possibly a new heart murmur on physical exam. This is how they would test your knowledge of strep viridans-induced endocarditis. Speaking of endocarditis, here's an image showing bacterial endocarditis of the mitral valve. We can see vegetations on the mitral valve, right here, from the bacterial infection. Do you remember which patients are more susceptible to getting endocarditis from strep viridans? Typically, this endocarditis is seen with patients who have previously damaged heart valves. For patients with previously damaged heart valves, we actually give prophylactic antibiotic with cefazolin before any dental treatment is undergone. Before we move on, what would you see on a gram stain of strep viridans? Remember, you would see gram positive cocci, but in this time you would see them in chains. In chains. Remember, Streptococcus species are gram-positive cocci in chains. And now thinking back to microbiology even further, how would you differentiate strep viridans from another streptococcus like strep pneumoniae? You can do the Optekin test and check for solubility in bile. Strep pneumoniae is Optekin sensitive and bile soluble, while strep viridans is Optekin resistant and bile insoluble. Very good. In the colon, we can see Bacteroides fragilis and E. coli. E. coli is the most important facultative anaerobe. These two bugs are both normal flora, although the number of Bacteroides is much more than the number of E. coli. And lastly, in the vaginal canal, Lactobacillus is normal flora. The vaginal canal can also be colonized by E. coli and group B strep. Do you remember what is important about group B strep? Remember, it can cause a meningitis and even sepsis in neonates. This is a pathogen that is always checked for in the pregnant mother by obstetricians to prevent transmission to the baby during childbirth. It is normal flora and usually doesn't hurt the mom, but it can be very deadly for the neonate. So speaking of babies, it's important to keep in mind that neonates who are delivered by C-section or cesarean section have no flora since they do not acquire it from the vaginal canal of the mom, but they are rapidly colonized after birth, especially if they are breastfed. Another step 1 favorite is to have you differentiate between the different types of bugs that can cause food poisoning. The time of onset of food poisoning and the symptoms of the food poisoning are the high-yield clues to correctly identify the bug. For the boards, they often ask you what food is associated with the food poisoning. One of the more interesting causes of food poisoning is Bacillus cereus. This is a spore-forming bacteria that survives cooking even at high temperatures. You'll typically see this food poisoning in patients who have just eaten reheated rice or pasta. Do you remember what the symptoms of this food poisonings are? This will cause a watery, non-bloody diarrhea with GI pain that usually develops within 24 hours. So remember, it is important to be serious about your Chinese food. Anytime you see mention of canned foods or honey on step 1, your first thought should be Clostridium botulinum. Do you remember what symptoms botulinum can cause? It can lead to a paralysis. Now, do you remember how does botulinum cause paralysis? It produces a neurotoxin, the botulinum toxin. And now can you remember what this neurotoxin does? This neurotoxin is a protease that cleaves a particular protein called SNARE. The SNARE protein is involved in the release of acetylcholine at the neuromuscular junction. So by cleaving SNARE, you do not have release of acetylcholine at the neuromuscular junction, and therefore we get paralysis. If you ever see dents in cans, or if you see bulges in cans, you should be thinking about botulinum. And do you know why these bulges and dents exist? It is because the Clostridium bacteria that are in the cans release various gases which manifest as these dents and bulges in the cans. Now in children, the boards will often present what is called a floppy baby. The baby usually has bad muscle tone and eventually can even struggle with breathing. This is typically the way they present infant botulism after the consumption of honey. Now, do you remember why does eating honey cause diarrhea in children but not in adults? Well, because the botulinum spores are found in honey, and they're able to survive, germinate, and even grow into toxin-producing bacteria in the gut of babies, but they are not able to in the guts of adults. The adults actually must ingest the botulism toxin to be infected. So the spores are unable to survive and grow into toxin-producing bacteria in the guts of adults. That's the key difference between the disease in children and adults. Reheated meat dishes and meat pies, such as the one we see here, can contain Clostridium perfringens. Do you remember the exotoxin that is produced by this bacteria? It produces an alpha toxin, which is actually what is called a phospholipase enzyme that degrades phospholipids. And this bacteria with this enzyme can cause myonecrosis, also known as gas gangrene, and can also lead to hemolysis. Undercooked meat can be responsible for harboring the E. coli 0157H7 strain, which is also called enterohemorrhagic E. coli, or EHEC, because it produces a toxin or an exotoxin that causes intestinal bleeding. This is very important. You will see a bloody diarrhea in patients with this E. coli species. Do you remember the action of this toxin produced by EHEC? The toxin is a Shiga-like toxin. It activates the 60S ribosome. If a child is infected with this bug and is also found to have anemia, what disease are you worried about? Remembering back to our pediatrics, we are worried about hemolytic uremic syndrome. Do you remember the classic findings that are seen in hemolytic uremic syndrome? In these patients with this syndrome, you are going to see anemia. You will also see thrombocytopenia, and you're also going to see acute renal failure. It might be important to know that renal failure is as a result of microthrombi formation, causing ischemia in the kidney. Now, if you saw a peripheral blood smear, what clue would you see that will help you think about hemolytic uremic syndrome? You would see the presence of schistocytes, and very good. So one final question, how do you distinguish this type of E. coli from other types? This species of E. coli, it does not ferment sorbitol. Undercooked poultry, meat, or eggs can all cause salmonella food poisoning. And what kind of diarrhea would you see in salmonella food poisoning? You would see a bloody diarrhea. So say a patient comes to your ER with nausea and vomiting after eating some potato salad at a picnic. What bug would you expect to be causing this food poisoning? Patients who have recently eaten meats, mayonnaise, or custard may be infected with Staph aureus. Anytime you read about a picnic or an old chicken salad, you should be thinking of Staph aureus food poisoning. Patients who get food poisoning from contaminated seafood are often infected with Vibrio parahemolyticus or Vibrio vulnificus. What is the other important Vibrio species that causes diarrhea? Remember, the other important Vibrio species is none other than Vibrio cholera. Remember that the Vibrio species absolutely love water. They cause watery diarrhea and are found in foods that come from water. Vibrio cholera infects individuals drinking of contaminated drinking water. So contaminated drinking water, as well as seafood, can cause infection with the Vibrio species. There's one more food association we've mentioned before that is worth remembering. Do you recall what bacteria unpasteurized cheese is associated with? That would be our friend Listeria. Remember that this is a particular concern in pregnancy, and it is why pregnant women should avoid unpasteurized cheese. Do you remember the first line treatment for Listeria infection? Ampicillin, very good. So now there are 2 bugs that cause rapid onset vomiting and diarrhea in roughly less than 6-hour incubation period. As quick as they come on, they also resolve very quickly as well. These are Bacillus cereus and Staph aureus. The reason for this is that the toxin is produced by the bacteria before you eat the food, and so you actually ingest the toxin along with the bacteria. Do you think antibiotics are going to help in this case? No, antibiotics would not help because the toxin is already made and has already been ingested, so the antibiotics will really do nothing to help with the diarrhea.